All right, so welcome back, everybody. So it's great to have David Redish here with us. David is a professor at the Department of Neuroscience, University of Minnesota. And he started life studying computer science and writing. That's correct, no? And that surprised me so much, because where I come from, we already get writing in primary school. So I was confused by that. Okay, just to annoy you a bit, okay? But we always make these lists for a school and for your company, like, okay, who are the speakers you really want to get? And David was on this list for a long time, and then we always failed, because we usually fail to realize many of our wishes. So it's such a great pleasure to have David here, because David has a sort of unique combination of a computational theoretical perspective on system level properties of brains and behavior, which is in itself rather unique, because most people like to delve into the details of their MATLAB code. But on top of that, he also actively gathers the data to advance and support these models. So this is really a unique combination of skills and insights, and I'm really looking forward to learn a lot from that. David, great to have you here. Thank you.
Thank you. Thank you. He actually, whoa, there it is. He actually set out the whole kind of history piece, and what I'm going to tell you about today is all the stuff that Tolman was talking about back in the 1930s and 1940s. In fact, I'm going to prove to you that Tolman was completely correct about vicarious trial and error. Before I get too far, however, I promised my publisher I would start every talk with a little plug. This is my recent book. It came out last summer. It's called The Mind Within the Brain, How We Make Decisions and How Those Decisions Go Wrong. And what it is, it came out from Oxford University Press. What it is it takes information processing and says, let's use our understanding of information processing to get at decision making, to really kind of open up the question of decision making. And I'm very happy to talk to people about this afterwards. I'm actually not going to talk about it today, because today what I want to talk about is I want to talk about cognition. And I want to talk about the work we're doing looking at how to measure cognition. I'm going to be very careful with my definitions in a second. So my talk today is going to be, I'm going to start by talking about this idea of what I call working in information space. And particularly from an experimental question, how do you actually use the data that you're collecting? I'm going to give a quick example, which is kind of a classic story, which is often called the word replay. I'm going to show it's not replay, but that's OK. And then I want to spend most of my time talking about this issue of deliberation. And I'm going to really prove to you, I hope, that rats actually do deliberate over choices. One of the keys here is that decision-making is more than just deliberation. We also have non-deliberative decision-making. And by following the information processing, we can make very specific predictions about the neural signals that we should see. And I will show you that those neural signals exist in the structures we expect them to be in. If we have time, depending on whether we have time, I'll present some new data we have, just came out about a couple of weeks ago, showing that rats represent the information about regret. Now, whether they actually feel emotional regret, we can discuss, but they have the information processing that differentiates regret and disappointment. We'll see if I get here. A whole bunch of people have asked me to add a section into this, so I did. Whether that means we don't get to this or not, we'll have to see what time goes. Please do interrupt me as we go. I'm happy to answer questions as we go. So first of all, I think the key here is to talk about technology. And I don't know how much technology you've actually seen, but this is, at this point, relatively standard multi-electrode recording technology in the rat. So here's a rat, and we implant electrodes through this microdrive into the animal. The electrodes are implanted permanently under, of course, sterile conditions, et cetera. And what we've then, of course, done is turned our rat into a cyborg, again, literally. I love that the science fiction terms are not science fiction anymore. And then we can record from the rat's brain as he runs around one of our mazes. I want to point out that, first of all, our electrodes are actually what are called tetrodes, which, of course, from the Greek tetra, meaning four. And there are 4 wires twisted together. And so each wire, that gives us stereo sound. A cell over here is going to sound different than a cell over here. And this is a recording from the striatum of a rat, and you can see very clearly 3 very differently identifiable cells that we can get from this one electrode, this one tetrode. We don't just have one tetrode, we actually have 12. The new technology we have in the lab has 24 tetrodes, but all of the data I'm going to tell you about today comes from 12 tetrode arrays. These tetrodes are individually movable, and so we can kind of optimize the cells that we get. The second piece is I think it's very important to think of mathematics as a technology, because just like a telescope or a microscope, it allows you to see things you couldn't see before. So this is the mathematical technology that we're going to use, and this is kind of the key to this story, and this is decoding. So here what we have is 30 place cells simultaneously recorded. Each circle here is basically an environment. It's an open field. It's neither actually open nor a field, but I didn't invent the task, so I don't get to name it. It's a big cylinder. And in the cylinder, the rat's just running around the floor eating food pellets that we throw in. And the point here is not really to talk about the task, but that you have these place cells. So this, of course, is a cell. This green is high firing and yellow is low. So this location, this cell likes this location, and this cell likes this location. And the key here is that if we know this relationship, then we can say when these 4 cells are active, we can decode the location of the animal. Okay? And the mathematics of this, when I'm giving a course like this, I like to actually spend a little time going to the math. The math of this is going to be a straightforward Bayesian update. Have you guys seen Bayesian decoding before? I don't know. Paul, have they seen Bayesian decoding?
Not maybe in tutorials explicitly, no.
Okay. So I assume everybody's comfortable with Bayesian probability that P of A given B times P of B is, of course, equal to P of B given A times P of A. And what we're going to do is we're going to take A and call it spike, or in this case, firing rate, and B is going to be some world variable. Now, I'm very careful about this. We're going to start with position. And one of the reasons, but it doesn't have to be position. It can be any variable in the world. And one of the reasons I like to come to that regret data at the end is we're going to use flavor for the regret data instead of position, but the math is all the same, and it kind of generalizes beyond this spatial stuff that I'm going to talk about first. But basically, we then have P of x given s is, of course, P of s given x times P of x divided by P of s. This is our decoding. The key here is that we actually know this parameter. This is the tuning curve. We have a prior, which we can assume is uniform, and we can produce other priors, but honestly, uniform is where we want to start today. And then a normalization. But the most important piece here is that we actually know this tuning curve. So the question then is, what happens if we decode to somewhere the rat is not? And this is a picture that was made for a science commentary where some other people were arguing over our data. So of course, the rats don't have electrodes. I love that Science magazine did not want rats with electrodes. But anyway, if you imagine a rat is running on some track, and then we take the rat off and put him over on a flower pot, and he's falling asleep. This rat, of course, is not asleep because we were taking pictures of it, and it was posing for us. But you could do decoding, and you see that you decode not to the flower pot, but to positions on this track. In fact, you get a very nice sequence of positions along the path of the track. Is this real? And that's the key question today, is I'm going to argue that this is real, and moreover, I'm going to argue that this is cognition. So this is a very old idea. This is actually data from Apostolos Georgopoulos in 1989, and one of the reasons I love this paper is that it's actually clearly about cognition, except he was not able to use the word cognition. And he told me that he wanted to, but didn't dare put it actually in the paper. So this is, of course, the classic monkey reaching task, where the animal has a manipulandum, and the animal has to hold the center, and then one of these other lights will light up, and the animal has to reach over to a light. If you record from neurons in the primary motor cortex, you see this very nice cosine tuning to the direction of movement. Of course, this is a lot more complicated. In fact, Stefano has done lots of the work on this and can tell you lots about how much more complicated this actually is. What I want to talk about is this decoding real quick. So the key is that with a unimodal representation, you can actually construct a decoding and get essentially the vector the animal is going to reach. So in this plot right here, the animal is starting at the center, is trained to reach to the light, and is just going to reach over to the light off to the horizontal. And you can see you get this vector of the reach kind of grows from the cue to the movement, and it kind of grows very nicely in the direction. But this animal was also trained that in another task, another block of tasks, when the light lit up at one point, the animal had to reach 90 degrees off. And what you see here is that this vector starts in the direction of the light, but then moves through the intermediate zones into the direction of travel. And the argument made by Apostolos Georgopoulos back in 1989, that's a long time ago, by the way. I wonder how many people were born in 1989 here. It's not, anyway. And what he argues, he points out, is that this is a representation, this is a mental rotation through that information. And of course, the interesting thing is the speed of this rotation matches mental rotation in humans. So how do we actually get to this? So let's take a look at the modern version, which I really have to say was due to Adam Johnson. I'm going to try to point out in this block right here, always this graduate student or postdoc who's doing the work, because of course, I just kind of get to come to Barcelona and talk about it. The fact is, they're the ones who are actually in the lab doing the work. So this idea, this is the mathematical technology. We have a rat running around some environment or doing something in the world. We get position in this case. We have some spike trains. I'm trying to use color as supposed to indicate different spike trains, but whatever neural variable. We're going to construct an encoding step, and that is, of course, the tuning curve. This is 93 cells simultaneously recorded. And then we're going to do a decoding step, which, as I said, if you have these 5 cells, of course, this is diagrammatic. Some of you may have noticed that I've grabbed the same picture here. I'll show you real data later. These 5 cells are active. We can do a decoding, and a decoding is taking the information from the tuning curve combined with information from spike trains to tell you a decoded location. There is, however, a third step here, and that third step turns out to be absolutely critical, which is if we know the location of the animal and we know the tuning curves, we should be able to predict the spike trains. And if we can predict the spike trains, then we understand this information. And the key here is that hidden within this tuning curve is a model of what we think the cells are doing. And so we can actually use this for model comparison. Go ahead.
I'm just wondering, you talking about rate-based earlier on.
Yeah.
This suggests that you're actually decoding for time-based or spike by spike based, rather than rate-based tuning curves. Is that right?
At this point, I haven't said anything about spikes versus rates. At this point, the idea is you take this information, you're going to construct some encoding measure. For us, it'll generally be rate-based encoding because that's easier to get at, but we'll get to specific spikes later.
Okay.
But the idea of this loop is independent of both the actual algorithm you're doing for decoding or for encoding, or for whether your spikes or rates.
I assume you couldn't generate spike trains from rate-based models. Could you have to have spike-based models to do that? Is that right or not?
This is actual data, so we have physical spikes.
Okay.
Yeah,
right.
We're not actually building computational models at this point.
Okay.
That's another talk,
Right.
which I could do, but not today. So if we follow, as I said, this idea does not actually depend on the specific algorithm we're going to use. Apostolos, for example, used this population vector model. We're going to use this Bayesian story for all of the data. And I want to point out that basically we're essentially taking the next step, which is we want to predict the spikes, or the rate, as a function of our hypothesized decoding, which of course is the decoding times our prior divided by, actually these are backwards, I apologize. This should be P of S hat given divided by P of X hat. But the important point here is we can measure this consistency by comparing this to our observed S. Right? And that consistency measure allows us to say this is not noise. And that's going to be key. I also want to point out, as I said, that there's really an M here hidden within this P of S given X, a little M here, because this is a model of what we think the cells are doing. So the argument here, to kind of complete this first step, is if we're decoding to a different location and this is a self-consistent representation, then this is evidence of non-local information. Okay? So let's take as our example the classic example of hippocampal replay. And I'm mostly doing this not really to tell you a lot about replay, but mostly to say this is something everybody's understood for many years and has actually thought of as non-local decoding. And it'll allow you to see how this mathematics allows us to actually get at that question. And then I'm going to basically spend most of my time, as I said, talking about the deliberation. So here are some place fields from actual data. This is from data from Anupam Gupta, who was in my lab. The gray dots are the locations of the animal. And I'm going to have a whole bunch of different tasks. I'll try to be careful about which task is which at each moment. So this task is what we call our 2T task. The animal runs up the central track and turns left for food or right for food under a complicated contingency, which I will explain later, but doesn't matter at this point. You can see very clearly there are these very clear place fields, which is the spiking occurs at a single location. Here's a nice place field. Here's another cell. This is actually from the same day. You can tell because the specific errors are all the same. What I want you to notice, though, is that on this place field, there's actually spikes occurring at extra locations as well. These extra locations are actually the feeder sites. And the rat actually spends most of its time at the feeder sites. If you look at the firing rate as a function of position, these are essentially 0, even though there are some spikes occurring. So the vast majority of the firing rate is occurring here, but there are extra spikes. And the question is, of course, are these extra spikes noise? The answer is no, and it's been known for many years. This is going back. This is a Kujamati paper I grabbed, where one of the things that happens is that when the rat is running through this sequence, the hippocampal local field potential is in this mode called theta, which is dominated by this 7 Hertz frequency. And in this, the place cells fire in their place field locations. In contrast, when the animal is at the feeder sites, the hippocampus switches modes. It's actually literally a state change that happens within the hippocampus. It's controlled by neuromodulators. And it's reflected as a change in the local field potential, where instead of having theta, you can see how much smaller the theta power is here relative to here. You have this broad spectrum thing with these punctuated 200 Hertz ripple events, sometimes called sharp wave ripple events, where these events happen and the cells burst in these ripple events. And I should, since John's in the audience, I should point out that the first person to show that if you actually decoded this position, the position at every moment, that when the animal was at these feeder sites, you got non-local decoding was actually a Jensen-Listman paper from, correct me, John, 2004? What?
The statute of limitations is over.
Fair enough. But anyway, that was the first paper that did it. I believe in actually giving credit to the first people who do things. So this is decoding. I'm going to show decoding at a spike level of the data from a single sharp wave. So the gray dots are, again, the positions of the animal. The black diamond is the location of the animal. You can see he's sitting at this first feeder site on the left. And here's the local field potential. Here's a sharp wave, series of sharp wave bursts. And the cells are colored from light blue to purple as a function of time. And you can see a very clear sequence going forward along this central part of the track. Sometimes here's a very long sequence going forward. Here's a backward sequence. Here's another backward sequence, etc. So even though you have these bursts are actually other places. So let's take this logic. We have some spiking data. We have some observable variable. You have some tuning curves. If we simply predict the tuning curves, when the rat is sitting at this moment, it should be in this purple tuning curve, this magenta tuning curve, and you should see spiking in the magenta cell. You don't. What you see instead is a sequence of spiking consistent with a replay or a play through the environment. Okay? That's what I just said. So this is, of course, exactly what everybody's been talking about in terms of the sleep replay. Here, instead of having, we have our observable variables, our tuning curves, and now the rat's physically on another location, what we see is a replay. This is just some data I grabbed from Matt Wilson's lab from a 2002 paper. What we see is sequences consistent with a representation in the other place. So the point here is that we have this consistent decoding about other places and other times. So to conclude this replay thing, I want to talk a little bit about the usefulness of looking at this in the information space. So not just that it exists, but what's the information within that. And this is Anupam's 2010 paper. So the training actually on this 2T maze, this is a 4T version I've plotted here, but this is the 2T you can see, is some days the animal had to go left, some days he had to go right, and some days he had to alternate. And on any day he would come in, he wouldn't know what it was, but the animal basically had to do one of these tasks, and then he would essentially learn to do that, or learn's the wrong word. He would recognize what the contingency is and then start doing that behavior. The reason I bring this up is that this rat would sometimes go left from this choice point, sometimes go right from this choice point, or left or right. He would never go back. And in fact, we went back. In the early days, when he tried to go back, we put a big stick behind him. By the time we're actually recording, he knows he's not supposed to go back, and he never turns around. But we went back, and this rat never went backwards on any experience this rat has ever had on this track. But here are backward replays followed by a forward replay. So the animal is here. It goes backward to the choice point and forward to the other side. This is a shortcut. This is a path the animal has never taken. Here's another one. Now, of course, I hope you're all saying, well, maybe you've just got two replays that you've shoved together. So how do we prove that? What we did is we reasoned that there were 32 possible combinations on the top and bottom rails on this track. 20 of them are disjoint. So for example, the bottom, this one plus this one would be disjoint. 8 of them are not shortcuts. They're basically out and back. And we're differentiating direction. So this is one and this is another. So there are 8 of those. And there are only 4 actual shortcuts. If we take this proportion works out to be this percentage. If we take all of the replays that we observed within any day and randomly shuffle them, what we find is this bootstrap, which is very much basically what we expect. If we take the actual pairs of replay that co-occur within 150 milliseconds of each other, I don't even need statistics. I'm sorry, that's not the same as that. There are no disjoint cases, and the vast majority of these are actually shortcut behaviors. These replays, these shortcut replays, are pulling up real shortcuts. This is a real event.
David, for clarification.
Yep.
Why would you call this a shortcut? Because in the task, in the left and the right contingency, the animal is not supposed to shuttle directly from left to right.
No. The animal is never, this is never a path the animal could or would take.
Exactly right.
Right.
So why would you call it then a shortcut? Because then it's not
It's a
task.
recognition that there is connectivity between this location and this location on this task. So it's a recognition of a novel connectivity within the space. We can call it, whether you want to call it a shortcut or not, I'm happy to say that. But the point is that it's a novel connectivity that the animal has never seen before. Go ahead.
Never traveled, it means in the same day, because it was experiencing that pathway on the other day, right?
It has never actually, because the animal never traveled backwards.
Oh, okay.
In fact, and that was why it was so important to go all the way back to the first day of training, which we have. We have, of course, video records from that. and the animal never actually traversed this path, going all the way back to day one. It's a very important
You never
question.
changed the mind.
What?
You never changed mind.
Changing the mind happens right here, and I'm going to talk about that next. But the animal was never allowed. If the animal went this way and tried to go backwards, we blocked him, and by the time that we stopped blocking him, he knew that that wasn't an option.
So can you say something about the density with which these paths are covered? Because actually, yes, the pattern is there,
yes.
But if you want the resolution varies quite a bit. So is there any systematicity to, let's say, a minimum, maximum kind of resolution you get?
Given the sampling of cells that we have within the very large, that would be tricky because we would have to work out the number of cells that we actually expect to be active. Of course, we're sampling from 100 cells out of, millions. Mm-hmm. So, yeah, I wouldn't be able to do that off the top of my head. We could probably work something out, but you would have to actually figure out what's the actual resolution within the hippocampal representation.
Your arm on that over lunch or so.
Well, I would need to go back to the data and actually work it out. Okay. It should be work outable. It should be possible to work it out, but you would need to basically translate how many cells are there within this part of the hippocampus, and how many cells are active at any moment, and sure. It's one of those things where I know the data pieces are in this paper, and that paper, and this data set, and that data set, but I don't know the actual numbers off the top of my head.
Okay. And similarly, for the relationship with this high-frequency response seems also vary, right?
No, it's right. The theory, 15 years, 20 years ago, was that these sequences were related to these high frequency. Turns out it's not. And that's a big shock and kind of issue in the field right now. I don't have an easy answer to that, but it's not. Okay, so what I want to really do, so the idea of that replay example was mostly to just show you the idea of using this technology to get at these questions. What I want to do today for most of my time is talk about this idea of deliberation. And one of the things I want to do is I want to take the idea of taking a psychological term, deliberation, and saying, how would we see this in an animal? So one of the big things, of course, is, rats don't deliberate over choices, right? This was why Tolman, in 1946, basically lost, right? If you take Psych 101 in any class, you will effectively not study vicarious trial and error in Tolman because rats don't deliberate over choices. But what does it actually mean to deliberate? What is the process of deliberation? So you go to the psychological literature, and they will say it is a search and evaluation process. That is, you are thinking about the future. If you're saying, where am I going to take my family for vacation? You say, well, we could go to the beach, and my son could go running in the water, and we would be able to think about, and you place yourself. You say, well, there'd be beach, there'd be sand, there'd be sandcastles, there'd be waves. Or you say, we could go to the backwoods of Minnesota, and we could go canoeing, and everybody would have a lot of fun going canoeing. And you essentially put yourself, you mentally time travel into that future, you construct that future, and then you evaluate it. Okay, well, at that point, I can look for it in the rat. Now, of course, the question is, where are we going to do that? And so we come to the human literature. We talk about this thing called episodic future thinking. And it turns out that, of course, patients with hippocampal damage are unable to remember their past events in their lives, to recall these past events. This is, of course, the classic HM story. Turns out these same patients do not imagine the future. If you ask them, what would it be like to go to the beach, they'll say something like, beaches have sand. Whereas if you actually talk to normal people about it, they'll say, well, I could take my girlfriend to the beach, I could go and we could walk in the sand, and I could feel the squish of the wet sand beneath my feet, right? And so you see this difference between it. This is beautiful data by Eleanor McGuire and Demis Hassabis in their 2007 paper. I actually love this plot. It's kind of one of my plots I always mention in schools that show the damn data. And the reason is, this is the patients. So these error bars are, these bars are different, obviously, but this patient didn't have that problem, right? What is quite clear is there is a deficit, right? But I don't want this number to pull this up. I want this number to recognize. I can see the actual data. I can see that this patient is fine and these patients are impaired. So, okay, so the key is that the same networks are involved both in this past memory and future thinking.
Can you say something about this experiential index they used there on that plot?
It was a description of the linguistic complexity of the description and how many relations there were that were in that, which kind of gets the whole relational issue. But it has to do with constructing the whole kind of environment in the thinking. So let's come back to Tolman. So as Paul showed very nicely yesterday, Tolman suggested this vicarious trial and error. Actually, again, I like to give credit where credit is due. The initial paper on this is actually Munzinger's 1938 paper. However, I think it's important to go back to Munzinger and Gentry, which is basically an abstract. And Munzinger later in one of his papers says, I really should have put Gentry on my 1938 paper, and I feel terrible about not having put her on it, given that it was particularly the complexity of being a woman in psychology in 1931. This is Evelyn Gentry and Carl Munzinger. But anyway, Munzinger and Gentry in the 30s, and they actually use the term. They're the ones who bring up the term vicarious trial and error. And then Tolman in 38, 39, 46, 48 proposed that it's really this imagination of the future. And then this vicarious trial and error vanishes from the literature completely. There are no papers. I have looked and looked and looked. I've talked to everybody in the field, and there are no papers that use the words until Hugh and Amsel come back in 1995. That's a 50-year gap. So Tolman is coming back, and he's coming back in part because we are after Claude Shannon, so we can talk about information. We are after Alan Turing, so we can talk about complexity in computation. We are after Newell and Simon, so we can talk about search processes. And we are after the whole neurophysiology revolution, so we have access to the brain. Before I show you what's going on with the brain, let me just show you, I'm going to show a little movie of an animal showing vicarious trial and error. Because, of course, vicarious trial and error is really about a behavior. I have a plugged-in animal just because it's a good example. If you want, I'll show you after the talk lots of examples of animals who are not plugged in. Obviously, Tolman's animals were not plugged in because he didn't have that technology. So the animal's going to run up this central track and turn left for food or right, left for food or right for food. There's little food sites right here and here. The contingency, again, doesn't really matter. This is just 4 laps picked out of a whole lot of laps. It's a pretty rare event. He comes up, he looks left, but he runs right. Here, he looks to the right, but runs to the left. On this one, he looks to the right, looks to the left. I don't know, man. Let's go right. And he goes right. I don't want you to think it's the cable swing here, because here the animal looks left, and when he gets to the choice point, he's already made his decision. Anthropomorphically, that's a good question.
Because his eyes are pointing upwards.
No, they're pointing, he can see, they're not pointing upwards. They're pointing, he can see down and around. He can see all the areas.
The Jason Kerr paper recently showed that basically they're monitoring space above their heads for potential failure.
To be honest, I have not actually measured that quantitatively, but I know for a fact that they can see cues both on the maze and...
Right, so there are a bunch of extra maze cues that
yes,
you think
there's lots of extra maze cues. There's lots of extra maze cues. But the extra maze cues do not inform you of the contingency, which is going to turn out. So the extra maze cues are not actually informative of the correct way to go. They're informative of what's on each side. But there are lots and lots of extra maze cues that are held constant.
What happens if you put them in an environment where there are no extra maze cues?
They actually still show vicarious trial and error.
Okay.
Well, there are never no extra maze cues. There are always extra maze. Well, there are always cues on the maze. I mean, there are local cues. There are odor cues. There are visual cues within the maze as well. Right. We've done this. I can show you later examples in tunnel mazes where the reward site is not visible from the choice point. They still show the same effects.
So you're not assuming that it's visual information necessarily which is triggering the
No, not at
brain
all.
cells.
Not at all. Although it probably is, but I wouldn't guarantee. We certainly don't have that. I wouldn't make that claim explicitly.
It would work the same in the dark.
Probably. I say probably because I'm trying to think if we've actually run any examples where I have in the dark. No, I haven't looked at it in the dark, but I believe it would also occur in the dark. But again, that's just from kind of anecdote of watching animals.
But that might be problematic if it would also occur in the dark, right?
Why?
Well, because then there's no visual information landmarks to obtain. So
Again, the question is what are they actually doing with the vicarious trial and error?
Yeah,
so let's go to that,
absolutely.
and then let's come back. So the problem is that the assumption that everybody's making, and the assumption when I hit this to this slide is, well, there's obviously some cue they're looking at in the future. I think that's completely wrong. I don't know if I can prove it to you that it's completely wrong, but let me go, and then we'll come back to that question at the end if people still feel we need to. So, yes?
Why you are considering the first turn as directed to the goal? Because there are no walls in the maze.
In this maze, I've done other ones.
So maybe the rat is just controlling the environment to not fall down.
What do you mean?
I mean, there are no walls, so the maze need to be well managed in the boundaries. So the rat is just checking if there is a floor.
No, the rats know this maze well. In fact, you don't see vicarious trial and error until the rats understand the space. So in fact, you have a transition. I always end up with this, where I basically give my talk and let this slide up. So the vicarious trial and error occurs after the animal understands the environmental space, but before he knows what to do on it. And so the animal knows that there's floors here very comfortably. The question is not what is in fact this way. The question is, do I want to go this way or not? And we know that from the timing of the vicarious trial and error. Okay, so let's take a look at what's happening with the place cells. So one of the things that's nice to notice, this is a very nice example. I should say, this deliberation story really comes from Adam Johnson's work. This data is from Anupam Gupta's 2012 paper. But Adam Johnson is kind of the one who came in and convinced me that this actually was real. And I have to say, I was extremely skeptical when he started, to put it politely. So one of the things that's interesting to notice about these cells is that this blue cell has extra spikes occurring at this choice point, and the red cell does not. So if we run a decoding operation of that, in the interest of time, I'm not going to actually run this movie, but we can see it right here. This is now a theta cycle, not a sharp wave. Right? This looks very different. This is now the 7 Hertz rhythm is active, and you can see gamma underneath, since John's here. And the black diamond is, again, the location of the animal. And now we see a sequence running from where the animal is to the goal site. And this is a vicarious trial. This is the position of the animal shown in blue. You can see the animal's running up. You got 1, and then this red diamond is this black diamond. The animal has come up. He's stopped at that moment, kind of throws this sequence forward, and then he's going to come back. And so what we have is this sequence of cells fires all the way, centered at the center of their place fields, all the way to the goal. And if we run a decoding operation, basically you get sequences going back and forth. It is important, as I mentioned, the animal is in theta. This is theta going through, and there are none of these sharp waves occurring. If this really is an episodic future thinking event, we can make very specific predictions about it. For one thing, we can predict that it's going to be serial. That is, you're going to go to one side or the other, not both. Because the key to episodic future thinking is that you're imagining that one future. You construct life at the beach, then you construct life in the backwoods of Minnesota. You don't do some sort of quantum hyperposition of the two. What we see is exactly that. If we look at the amount of probability distributed ahead of the animal, it is either left or right or local. There's nothing in the joint component. If we look at, is it ahead of the animal or behind the animal, by basically taking how much of the probability is decoded ahead of the animal versus behind the animal, we see it is significantly ahead of the animal, not behind the animal. These are, and this of course is what we'd expect from that blue cell. These are representations going ahead of where the animal is to the choice point. So that suggests that there is actually a reflection of that search process. Now, if...
David,
yes,
have
go
you
ahead.
looked at the timing of this process relative to the head turn?
Yes. So what you actually get are the direction of sweep is probabilistically related to the head direction, but it's not 100%. So it's about 80% the direction the animal is going to go. You will often see, we have not quantified it yet. It's one of these things of getting the students to actually run the analyses. We have seen many, many anecdotal cases of where the animal comes up, stops at the moment, you see sequences that way, a sequence this way, and then he comes back, kind of right at that moment, within about 200, 300 milliseconds. But certainly we see sweeps in in each direction, independent of head direction, as well as probabilistically related to the direction.
That was my question, because rather than necessarily collecting visual data, he could just be turning his head in order to fire head direction cells, which would then initiate this computation.
It is quite possible. And so one of the interesting questions is, how accurate are head direction cells? One of the things that's been a big issue with head direction cells is, if the rat runs backwards, do the head direction cells point forwards or backwards? Right? There is new data coming from Drew Maurer in Carol Barnes's lab where they've got animals to run backwards. To be honest, I don't know what the head direction cells are doing in that. I believe they have the recordings. Michael Zugaro has a train that runs backwards. We know that the phase precession, which I'm going to talk about in about 2 seconds, runs backwards on those tasks, which is that the phase sequences are in the direction the animal's going, not the direction the animal's facing. Whether the head direction cells flip sides is a very interesting question, but we don't know.
Maybe not specific to head direction cells. I'm just thinking, if I'm trying to imagine what's down that street, that helps me to kind of, even if I can't see, to orient myself as if I was facing that direction.
I agree completely. And I think that's a lot of what's I think actually that's what the vicarious trial and error behavior is. I think it is exactly that, which is the initiation of a movement so that you can kind of throw that sweep forward. Now, it is possible, just like all those monkey experiments where they force the monkey to focus fixation and then they do stuff over here, even though that's highly unnatural. And I don't know, I always find it impossible to do because I want to focus and foveate, right? It is possible to hold yourself fixed and do stuff. And it is possible, we will occasionally see animals do this. Hugo Spears, who used to be in Ellen McGuire's lab, now has his own independent lab at UCL, has actually done human stuff kind of like this. And you can force yourself not to, but the natural behavior is very much to kind of initiate the motion, go, wait, that's not what I want. So I agree. I think a lot of the behavior of vicarious trial and error is, in fact, that initiation of motion. There was a question. Did you have a? Okay.
Well, I have one, David. So what you show is that you have fidelity to the choice, right, in the previous slide. But what you haven't told us is how accurate the elements of the forward sweep are. For instance, to what extent do you possibly switch place cells in the order of their recall? Like, what's the fidelity of the exact sequencing of this forward sweep?
I don't have a slide on the quantification, but this is as forward as any sequence you'll see in hippocampus. It is always forward. It is never backwards. Every theta cycle we've ever seen has always been forward. We've never seen backwards.
But now, if you look into, you see this little clustering in the center.
Yes,
So
I don't know.
My question then is the order in which these cells are firing still showing fidelity to the actual behavioral sequence, or is there now, let's say, some noise entering in that forward sweep?
The noise within this forward sweep is no different than the noise we see in any other theta cycle sequence. What the fidelity of that is, I don't think we've quantified, but it is it is certainly not random. And if we were to, for example, we actually control the way we define a forward sweep is we compare it to shuffled spike ordering. And basically, this is vastly not random.
Sure, no, I believe that. But for instance, if you
Yeah, I don't know how
it relates a bit
specific
to
the
Stefano's
individual.
talk, right? Because
Right.
in Stefano's case, you anchor to the center of a sequence. Here you could argue I might anchor to my
No,
feeder site, or
it's
I might anchor to where I am.
It's anchored,
Right.
there's no question in my mind, and in fact, I'll show you data in about 3 slides. It is anchored to the beginning and the end.
Right.
It is not anchored in the middle.
But it might then mean that this forward sweep says, look, I'm pretty sure about the beginning, I'm pretty sure about the end, and in the middle, it should sort of be like this.
It could be. I don't think so. Anecdotally, thinking of the many, many cases I've seen, I haven't quantified it. But absolutely, it's much more anchored at the beginning and the end. So I want to now kind of talk about disruption of the hippocampus. What is the hippocampus actually providing to deliberation? Before I do that, I need to take a side note. I was going to do 1 slide on this, and after, as I said, about 8 different people asked me to put in this new data, which is not published, and so I'm going to spend about 4 or 5 slides talking about a very exciting new result that we've got. But before I get there, what I want to do is mention this idea called phase precession. So this was first observed in the 1990s, 1993 by Redish and O'Keefe, and then by Bill Skaggs, did a lot of work in the 96. This is just a paper from Jimin Shen in 1997 I happen to have access to, so it kind of gives an easy thing I can throw up on a slide. The animal is running up, going through this place field. The color of the dot is the phase of the theta cycle that you're seeing firing. So if you plot position on the track through this place field against the color, against phase, what you see is this kind of shift, and that's what this color is showing. And one sec. So what you actually have here is the theta is moving, and you see the spikes occur at a specific phase of theta, and you can see that it changes through.
I was going to ask phase relative to what?
Relative to the theta cycle recorded from the local field potential.
Okay, so you have a reference, and that's stable. And
Well, the theta cycle is stable. You can cut it at basically whether you cut it at trough to trough, which is where the theta if you look at where the place cell, the pyramidal cells in hippocampus fire, they will zero out at a certain moment. Call that the zero of the phase. That's going to come out in this case at the peaks of this phase, because this is recorded from the fissure. If you record from the layer, it'll be the trough. But you basically say, I mean, theta is a sine wave, pretty close.
But it's completely stable. There's no noise or jitter in terms of the frequency.
There is some noise, but you're talking about a difference from 6 to 8 hertz, and it's relatively.
So that in phase terms, that would make quite a difference in terms of this.
Yeah, but we actually define phase as peak to peak. So we're going to just define phase as peak to peak that way. The subtleties of actual theta shape we could talk for hours on. Well, let's leave that off for now. So what I want to do, though, is I want to talk about this. This is work by Andrew Wickenheiser, which was his thesis he just finished, and the paper is under review right now. And what I'm going to show you is that these theta sequences are goal-related. And that's this side note I'm going to spend about 4 slides talking about. So on this task, the animal's running in a circle. He's got 3 feeder sites, each of which is going to provide food after a delay. The delay is completely cued. There's a tone that's going to count down, and the pitch of the tone tells him how long he has to wait for food. So as the rat approaches a feeder site, obviously there's going to be some divergence where he's going to come and stay at the feeder, and some where he's going to leave. So we have to kind of mark that divergence point. I should point out this is behavioral neuroscience. We always like to know the animals are actually running the behavior. So they are sensitive to delay, skipping long delays, which makes sense. Now on this task, unlike some of the later tasks I'm going to talk about, the delays are constant within a day. So today it might be 4 seconds, 8 seconds, and 20 seconds. Tomorrow it might be 6 seconds, 12 seconds, and 30 seconds. But it's constant within a day. So once the rat has done 1 loop, theoretically, the rat knows what the delays are. Okay? So we get place fields on this task, and we get firing at the feeder sites, just as we would expect. We can think of this task as dividing into 3 kinds of loops: 1-step loops, 2-step loops, and 3-step loops. Okay? And the key here is that we have not told the rat when to take a 1-step, a 2-step, or a 3-step loop. It's entirely the animal's choice. Now, one of the issues, which again I don't want to spend a lot of time talking about, is theoretically, optimally, the correct thing to do in actually every single case is to always just take the shortest delay, run a full loop, and wait for the shortest delay, and run a full loop, and the animal will get the most food that way. That's not what they do. There's a lot of very interesting, weird aversions to kind of skipping a food offer that they do. That's a whole other issue. It's kind of the economics of it, which we can talk about later. For now, I just want to talk about these different steps. So here is an example of a rat running 2 steps. And what you can see is 6 different examples of phase sequences. And these are colored from yellow to red, where what you see is a sequence. And yes, I agree, it's not completely perfectly ordered. Whether that order is noise, how much of that noise is there, I don't know. But you can notice that it generally starts kind of around the beginning of this journey, right, where he is, and it ends every single time at this feeder, at the feeder he's going to end up at. This is intriguing. This is not data. This is data, but it's not, quantitative. So let's be quantitative. Let's measure the look-ahead distance. Now, we have some controls we have to put in. One is we're going to rotate the maze, so the animal's always starting from here. We rotate, the analysis, not the actual maze. We rotate the analysis, so the animal's starting from here, and he's going to go 1 step, 2 steps, or 3 steps. And we're only going to take the zone in that first component that is identical under all 3 conditions. And we've checked to show that there's no effect of speed, there's no effect of acceleration, and there's no effect of which starting point you actually have picked. I don't have those controls up. I can show them to anybody who wants afterwards, but none of those have an effect on these results I'm about to tell you. But what I want you to notice is that the 1 step is much shorter theta cycles than the 2 step or the 3 step. There's a clear trajectory. The farther he's going to go, the farther ahead of the animal you're seeing this sequence. We can do the same analysis arriving at a feeder. So now we've rotated everything, so he's arriving at this spot, and we're only going to take the final trajectory, which again is the same under all 3 conditions. And here, there's no effect at all. In fact, when we look explicitly at this, what we are seeing is that it's going from where he started on the journey to where he ended on the journey, and it's goal dependent. One of the very interesting predictions of this is that the place field should be of different sizes, because there's a very clear dual relationship between the place field you're going to record and the sequence that you're observing. So what we're seeing here, if I can, let me just take a moment to go through this plot. Each line here is 1 lap. This is kind of 1 start. I don't, let's say it's here, and this is the end. And we've obviously chosen this so that the place field's kind of in the middle. This is a single cell. The blue dots are 1-segment cases. The red dots are 2-segment cases. Notice how they both end at about the same point, but their starting point is different. And in fact, if we measure the center of mass, there is a pair of parallel lines. The fields are starting earlier and they're ending. The cells are all about, I am coming to this point, which for the record was an argument John Listman made in the 1990s, and we all said he was crazy. And I love that because he was right. This is an example of, of another cell, which again has 1 step in blue. There's a couple of laps of 1 step, a lot of 2-step laps, and 3-step laps. And you can see that the field changes completely on a lap-by-lap basis. What this tells you is this is not that there's a modulation coming in on a lap-by-lap basis relating to the goal of the animal. These trajectories that we're seeing are directly related to the goal of the animal.
Is it constant? So if you look at this time difference in black and red and blue, which is always a 1-segment difference, is the time window also constant if you compare the 2? Like if you look at the onsets of blue and red.
Yeah, I haven't. That's a good question. We haven't looked. This is just 2 examples.
Sure,
I'd have to look at lots of them. I don't know. That's a good question.
What would your prediction? Would you expect that?
Yes.
Okay.
I would predict that, but I don't know that answer.
Are you measuring the number of loops of the theta, right?
No. What this is just the fields as a function of position. What this stuff is the length of one of these segments from the first cell that's recorded to the last, sorry, the first cell that fires to the last cell that fires. That's what these lengths are. And this is single cycles of theta. So this is a single theta cycle that we are, basically we're saying, here's a theta cycle, and we're going to start from here and go to here. We're going to say, what's that length? That's what we're measuring. So what I want to argue, and kind of the conclusion of this side note, and a few other data points, which again I don't want to go into details on, but I could, is that what's really going on here is not phase precession. Phase precession is a consequence of this sequence plus linear motion. And so if you have a stable position, for example, you see the same sequence over and over and over again. If you have mental progression, you do see a sequence. If you have bidirectional, going one direction or the other, the sequences go in the opposite directions. And if you have this deliberation story, what you're actually seeing in this kind of vicarious trial and error, really ought to be called VTE, not deliberation, because I haven't proven that it's deliberation yet, is you're going to see, the right side, then the left side, and this is, obviously an extreme, but we've definitely seen single cases where it goes to one direction and then the other and then back to the first. And that tells us that the sequences are happening on a theta cycle by theta cycle basis.
Yeah, so that was my question, David, but maybe you're going to deal with that.
Okay.
So this is a beautiful data set, but doesn't it actually invalidate your notion of deliberation?
It doesn't invalidate note of what?
It invalidates your notion of deliberation, because what you show us is that the animal is basically telling you exactly where it's going to go. It's not deliberating anything. It's just going to go
not on
there.
this task. Not on this task.
Exactly. That's what I'm saying.
No. On this task, absolutely, the animal has already made his decision.
Exactly, right. That's
right.
the point.
But on the multiple T task that I showed you earlier,
Sure.
then he's deliberating.
Exactly. But
No,
I'm saying,
absolutely.
But this data set might not help you then to make the deliberation point.
No, it doesn't.
Okay,
The deliberation
agree.
point is from the T case. The reason I wanted, so partially the reason I brought this in is because, as I said, a whole bunch of people have been asking me about it. And when it shows you that it actually relates to the goal, that the phase sequence relates to the goal. The real reason I wanted to bring up phase precession was because I want to bring in David Rob's data. And the reason I want to bring in David Rob's data is he has this fascinating cannabinoid result. And that's going to turn out to really inform us what hippocampus is doing in deliberation. So this is David Rob's 2009 paper when he was with Bujaki. He now has his own lab. I believe it's in Marseille. And what he found is he basically gave a drug that is the equivalent of cannabinoids, of marijuana, of pot. And what he found is that the place fields remained relatively constant, but the phase precession disappeared. Now, he doesn't have sequences, he only has single cells, but as I showed you, the phase precession is indicative of the sequence. And the sequence is what we're talking about with deliberation. So the phase precession being gone implies that there's a sequence problem. And this was the key figure for me, which he does not talk about vicarious trial and error, but I know how to recognize vicarious trial and error. This is a task in which the animal is alternating sides. It's basically a working memory task. So he picks some side, he has to come up and then decide whether, alternate to the other side. Here's an error where he goes to the same side and where he alternates. This right here is vicarious trial and error. The animal comes up, he looks to the left, changes his mind, and comes right. That is, I don't even need to, I've talked to David about that, and he goes, yeah, that is exactly what it was. But I can see from this data point. What's interesting is that under drug, you have a dramatic increase in the amount of vicarious trial and error. Actually, messing with the hippocampus increased the vicarious trial and error, which was not what I expected. And it goes away when you wash it out with recovery. So we've actually quantitatively started looking at, where am I going with this? Why am I doing this? Oh, right. Now I know why. So the other piece, so point one is that hippocampus increases vicarious trial and error in David Rob's data. We're going to he wasn't quantitatively measuring VTE. We're going to quantitatively measure it. We're going to use it doing this integrated angular velocity. Details aren't important. I can go through it if you want. But basically what we've got is sometimes the rat goes right through, and sometimes he does these, turn events. And we're going to take this and we're going to get a essentially a skewed distribution. So you get the VTE events are way out here on this distribution, and you get this what is effectively a Gaussian. This is not a lot of data points. When you get lots of data points, what you get is a Gaussian that shows not VTE and a skew with a tail that shows VTE. And so the amount of this tail tells us how much vicarious trial and error an animal is doing behaviorally. This is just on the task that I'm going to actually show you the data from. We call this our restaurant row task. If I get to the regret data, we'll talk extensively about this. Here, a rat is running around a circle and has basically 4 offers of going to different flavors of food. And the important point here is that he has 4 choices where he has to make kind of left or, where do I go? Sometimes he just skips because it's not a good deal. Sometimes he takes it because it's a good deal. And sometimes he kind of looks in, says, I don't know, man, all right, fine, and then goes on. And we can do this same kind of analysis where we find that the vicarious trial and error events are way out here on the tail of this distribution, and the just pass through, whether he's passing by or going in, are in the main section of the distribution. And the reason I bring this in is because now we're going to start to do disruptions of brain structures. And I don't know how many of you have seen the new kind of optogenetic DREAD kind of technologies. We're using DREADs. Have they seen DREADs? Nope. Okay. So again, I love this. This is science fiction. This is mad scientist territory. Okay? We are manipulating the brains of animals to do this. That's just the coolest thing ever. So this is a viral technology developed by Brian Roth. And what he does is he has a genetic construct put into a virus. That virus changes the DNA, which changes the proteins, which changes the receptors, which of course changes how the cell responds and how the cell computes. So what this receptor is a pharmacological receptor to clozapine N oxide, which is inert in the rat. So you take a normal rat and you give them clozapine N oxide as a IP injection. Nothing happens. The rat doesn't care. There's no difference between doing that and giving a saline injection. And in fact, once rats are used to it, it doesn't affect their behavior at all. But if we have this viral construct and we put it into a part of the brain, now that brain responds to CNO. We do just the viral construct. Again, the rat doesn't do anything. We've added a few receptors to a couple of neurons to something that doesn't exist. The amount of extra proteins that's been generated just doesn't do anything, right? Again, that rat has no behavior, no change in behavior. But take a rat that has the virus in that part of the brain and give that rat CNO, now we have pharmacology. And so we can create pharmacology. And of course, because the whole DNA world these days is this big if-then control structure, we can say only in these cells that have this protein that are doing this, then we're going to only generate that in those specific cells. It's the coolest thing ever. So we've now got this working, and we're going to do this in prelimbic cortex. I have some hippocampus as well, but I'm not going to show it because the data is too preliminary, and I don't have a good... I'm going to have to use the David Robb data to defend the hippocampus at this point. I'm not comfortable with our DREAD data in hippocampus yet. It's still too messy. But our DREAD data in prelimbic is very nice. You can see these as a CAMK2. We're picking up only the pyramidal cells in the prelimbic, which is the prefrontal cortex of the rat. And what you can see is there's a clear decrease in the CNO condition in this skewed side. We've shifted this specifically. Now, a nice control here is that we've also done this with orbitofrontal cortex, which has other effects, but it doesn't have any effect on the vicarious trial and error. So CNO alone is not doing this. It's a function of the manipulation we've done in prefrontal cortex. John?
And what's the axis on that?
The axis is this skewed log ID phi is a measure of the vicarious trial and error. And the key here is that the non-VTE events kind of live in this Gaussian down here, and the tail is the vicarious trial and error. So we're looking at the shift in the tail. Does that answer your question? I mean, we can go, the ID phi is actually integrated change in angular velocity, which turns out to be a very good measure of that stop and look behavior.
Are you saying that less vicarious trial event sweeps or less
No,
behavioral?
less behavior.
Less behavior.
All we have is behavior. We also have the recordings, but as you know, it can take months to get the data and cut the cells, et cetera. So we have the data, but I have no idea what it does. And I'm really excited to find out, but I don't know. So no, this is pure behavior. But what this means is that the way to understand this prelimbic hippocampal interaction is that hippocampus, the prelimbic is sending some sort of what would happen if message, let's go tell me what's going on. And hippocampus is responding with this is what we find. And so the David Robb data, particularly the cannabinoids, is coming back saying, well, I don't know, man. Right? So the prelimbic says, go check for me. Hippocampus comes back with garbage. Prelimbic says, that wasn't the question I asked. Go look again. And we get an increase in vicarious trial and error. On the other hand, if we knock out the prelimbic with DREADS, what we get is no request. And so, in fact, we see a decrease in the vicarious trial and error. And that suggests that search is actually initiated by the prefrontal cortex and enabled by the hippocampus. And this is consistent with what the humans are seeing. Now, of course, a very interesting question is, what about that evaluation process, right? If this really is deliberation, it's not enough to say I have an expectation of my situation. I have to evaluate it. So the obvious place to look is in the ventral striatum and the lateral orbital frontal cortex, which are the areas known to be involved in motivation and evaluative steps from all the neuroeconomics literature. So this is data from Jeff Stott. I have data from Matt Vandermeer and Adam Steiner I'm going to show in a few slides. This is actually part of that philosophical transactions. I think it's the one that Paul was editing. He says he recognizes it. So this data is, this is ventral striatum in blue and orbitofrontal in red, and you can see these cells respond to reward. This is a slightly different task. In this task, the animal has to go up, make a choice of taking a large reward after a delay or a small reward immediately, and there's some contingency complications that I don't know if I'm going to end up needing later. If I do, I'll get to it later. The important point is we have this same kind of single T choice. But what we have here is you can see this is cells that respond to reward either by increasing or by decreasing their firing rate, and so we can take the information from that and we can ask questions about it. So this is Matt Vandermeer's data from the multiple T maze. This is the maze that was very much like that Anupam Gupta maze I started with. The animal's running up the central tract to turn left for food or right for food. And what you find here is that you have reward cells. Of course, the spikes those cells are going to fire are going to occur at the reward site. And these cells fire a few extra spikes right at the choice point on those early laps where you see vicarious trial and error on this task. And if we did a decoding operation, now we're going to do a spatial decoding operation with reward cells, which is kind of tricky. But what happens is that if you do that, the vast majority of the spikes are occurring at the reward site. It's going to drag your spatial decoding over to the reward site. It's basically a way of using the spatial decoding to get at reward questions. We'll do real reward decoding later. But what you can see is there's a representation of the feeder site that occurs slightly before the animal does its turnaround moment. So this time right here is the moment when the animal has stopped before coming back. This is ventral striatum. This is Adam Steiner's data, which came out in Frontiers. I should mention this was in, they're both Frontiers. So this is a 2009 paper. This is a 2012 paper where Adam Steiner looked at orbitofrontal cortex doing this exact same task. This is actually a left-right version of this task. But here you have very clear data. Of course, we have better kind of cameras at the time, now that we know what we're looking for. So here's an animal coming up, coming to the choice. This is the choice point right here on this maze. He comes up, he stops, and he comes back. And notice this cell, which is a reward cell, it fires most of its spikes at the reward site, fires pop, 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 1, 2, 3, 4 spikes, just as the animal turns away from that moment. The animals come up, he stops, and then it's 1, 2, 3, 4. And if we do the decoding is after this turnaround moment. That's very different from the ventral striatum, which was before the turnaround moment. Now, I hope everybody's unhappy with this. This is 2 different students, 3 years apart, 2 animals on slightly different tasks. This task is only one choice. There's a switch. There's flavors on this one. It's not identical. So Jeff Stott went back and actually analyzed on this task simultaneous recordings between the ventral striatum and the orbitofrontal cortex. And I don't know how many of you actually do behavioral neuroscience, but it's so rare to see things really replicate perfectly. This is about as good a replication of the ventral striatum. Notice it's before. Here's your burst, just like, and the orbitofrontal cortex afterwards. That's just beautiful. So this really is that ventral striatum is representing the reward before the turnaround moment, but orbitofrontal cortex is after. So what this suggests is that there's some sort of prelimbic is initiating this deliberate event. The hippocampus is constructing this episodic future. Both of these are somehow projecting to the ventral striatum to do the evaluation step. To be honest, I have no idea what orbitofrontal cortex is, what role OFC is playing, but it can't be the evaluation step because the animal is actually already turned back by the moment that the orbitofrontal cortex is coming in. I think it's some sort of expectation game. We can talk a little about that later. How am I doing on time?
Great.
Okay. Good. Then I will just keep rolling. So,
Now look, where are we in your...
I'm right here. Actually, this is great.
Okay.
Yeah.
Now we're doing well.
Okay. Yes, absolutely.
I'm getting a little confused. When the animal is in free play,
Mm-hmm.
forward sweeps,
Mm-hmm.
it's seen in, you said it is in theta.
Correct.
But is it true that it has also shown in ripple?
No. So the ripples, there are, okay. So there's a debate between myself and Lauren Frank, who has argued, Lauren has argued that he's seen ripples at the choice point. I don't think they're ripples that he's seeing. I think they're a high frequency gamma oscillation. Our data is absolutely clear that the animals are in theta. And it does not, the animals are not in LIA, which is the non-theta that drives ripples. One of the really interesting questions is there's some data from, among other people, David Foster, that the ripples, that you'll see ripples before an animal takes a trajectory. And actually, I have a student right now who's looking on this task in hippocampus. And you absolutely, of course, see sharp wave ripples at the feeder sites. And we definitely are seeing kind of ripples before the animal goes. And the question that I don't know, and I want to be careful, I don't know the answer to this, is there a relationship between some sort of forward kind of ripple sharp wave plan and VTE? That is, when the animal does vicarious trial and error, is there basically 2 ways to make decisions? One is, I know where I'm going to go, I'm just going to go. And the other is, I'll figure it out when I get to the choice. And whether there's a relationship there or not is actually something we're chasing right now. I don't have that answer. My gut says that basically they're going to be anti-related, but I do not know that answer yet. Does that answer your question? Good. Okay. So I want to now talk about the other side of deliberation, which is automatic habit behavior. So, actually, I do have to talk about this task. So this is work by Andy Papali and then Jeff Stott. So we have this task. Now, what this task actually does, this is a neuroeconomic task. And both for this and then for the regret data, if we get to that, we really have to think about it from an economic perspective. So the animal here has a choice to go take the large reward after a delay or the small reward immediately. The large reward might be 3 pellets after 6 seconds or 10 seconds or whatever. And the way this task works is if the animal has gone to this large site, he's telling us the large reward after this delay is more valuable than the small reward immediately. So, okay, I'm going to make the large reward after the delay worse. I'm going to increase the delay on the large reward. On the other hand, if the animal has gone to the small reward side, he's telling us that the small reward is better. I'm going to make the large reward better. I'm going to decrease the delay. So now we have basically a Mazur-type titration task, but translated into space. And it turns out that because rats love to alternate, they actually show, this is real data and I've drawn some lines on top of it, this is that the rat actually does exactly what you'd theoretically expect it to do. It spends a couple of laps alternating which basically holds everything constant. And this is figuring out for this day, how many pellets is it getting, because obviously we change it from day to day. What's the starting delay, because again, we change it from day to day. Which side is the delay, etc. Right? So it says, oh man, it's a 29-second starting delay for 3 pellets on the left side. Forget it. I'm going to go to the small side for a while, which will drive this down, and then it's going to alternate to keep the exploitation. Now, that's a whole anthropomorphic story. What I'm telling you is, in fact, the behavior shows this investigation, titration, exploitation behavior, whether it's going from top down or too short, where it then takes the larger reward side for a while to drive it up. Okay? The interesting thing about this is that if we look at the non-VTE laps, that is laps where the rat just blasts, I don't know if you can see this, he's done black on black, which is hard to see, but if he blasts right through here, then the case, it's almost always an alternation lap. That is, it's almost always either an exploitation lap or an investigation lap, or it's one of the cases in the titration where it's alternating. On the other hand, in the cases where there is a VTE event, it's 50-50. You cannot predict where the rat's going to go from the starting point. So if I'm here and I know that on the next lap he does not show VTE, I am almost positive he's doing an alternation to the other side. On the other hand, if it's a VTE lap, I have no idea which way he's going to actually end up. And this tells you that the vicarious trial and error behavior is a flexible behavior. It's a behavior where you can change your mind at that moment. But the non-VTE is some sort of more automated, less flexible behavior. If we go back to the multiple T lap, this is actually work by Matt Vandermeer reanalyzing his data, Adam Johnson's data, and Neil Schmitzer-Torbert's data. We can look at the choice point and compare it to the control box. Now, this is from a while ago, and so we weren't looking at VTE. We were looking at time spent, which of course turns out to be highly correlated. And what you can see is the proportion of time spent at the choice point and at this topologically identical control box is on the early laps of this, when the rat doesn't know what the contingency is for the day, spending a lot of extra time at the choice point. But as the animal automates its behavior, this normalizes out to basically be that he's running at a constant speed in both conditions, or through both boxes. If we look at the hippocampal data, that is primarily in these early laps, that's where you're seeing these sweeps to one direction or the other. Once we're back in this zone, those sweeps have pretty much gone away. Similarly, if we look at the extra activity at the fourth choice point, the extra cells, the blue are the reward cells, the red are the non-reward cells, so this difference between at the feeder site between reward and non-reward is a definition difference. This blue blip right here is not. That is real data. And what is happening is the reward cells are firing extra at the fourth choice point. That is only on the early laps. It goes away with late laps. So we have some sort of transition from this deliberative kind of behavior to something else. Well, the obvious something else is Hull type, Clark-Hull type stimulus response behavior. And actually one of my favorite points is in the big Tolman versus Hull debate that Paul brought up. They were both looking at rats making decisions on T kind of choice mazes. But Tolman said, well, I'm really interested in learning. I want to know the beginning. That's when all the interesting stuff's happening. And Hull was, well, at that point, everything's variable, and I can't analyze my data, so I'm going to wait till everything basically stabilizes. And it turns out when you look at Tolman and Hull, Tolman's looking at the early laps, and Hull's looking at the late laps. And that turns out to completely explain the differences that they saw in their data. So if we're going to talk about this, we're going to talk about some sort of automated motion. And in fact, my examples, I think the best examples of this are sports. These are behaviors that you have to train yourself up to do, but you really don't have time to deliberate. Don't forget, the main point of deliberation is it's flexible because you're imagining the future. But if you're trying to hit a baseball or hit a triple axel jump, you don't have time to say, well, okay, if I put my foot this way, and if I put my foot that, you do that, you're on your backside. Right? You have to have this trained up that you can basically store the action chain and release the action chain. Okay? If we buy this, we can make very specific predictions. First of all, The cells in this structure should represent key situation parameters, and only the situation parameters that matter for the task at hand. We should not see these expectations. We should not see sweeps like hippocampus shows. We should not see reactivation of reward cells like ventral striatum shows in deliberative behavior. What we should see is we should see these situation cells develop with experience. Now, for various reasons, which I don't have time to go into, but one sec, which I will, I can go in if people need me to, is we looked in the lateral dorsal striatum for these results. You had a question? There was a question.
You just said that you wouldn't expect to reward the spikes on the edge, but would you expect it on the orbital fundal?
Ooh, that's a good question. Yes, I would. You actually see them in the ventral striatum. And in fact, you see there's a whole kind of second aspect of ventral striatal cells that turn out to learn to predict kind of slowly these ramps of activation, which are probably related to value ramps. And those seem to develop with time. So I do expect to see that. My point was not so much, and in fact, we see reward cells in the striatum, in the dorsal striatum. And I'll show, that monkey people see that, we see that. I'll show you that. What we don't want to, what I predict should not happen is that the reward cells should not reactivate in those at the fourth choice point. So, first of all, this is a task.
Are the ventral striatal cells ones that receive hippocampal projections?
The ones that reactivate? Almost certainly. We don't know. I haven't done the simultaneous kind of triggering, but it's from that area, that part of ventral striatum. And we know from Cyril Penartz's work that during sharp waves, the ventral striatal reward cells will reactivate in relation to hippocampal sharp waves, and they will time lock to the hippocampal replay. We also know from Matt Vandermeer had an analysis where he looked at ventral striatal cells, actually the non-reward cells it turns out, but relative to hippocampal theta, and they phase processed relative to hippocampal theta in very interesting ways. So we know that there's a hippocampal influence on that.
So David, why are you so strict on not wanting to see reactivation of reward cells? Because you could argue that you might need to, for instance, to maintain consolidation of the procedure of the habit.
We'd have to go through the information processing algorithm of that consolidation. What I want to say is during the moments that you would expect during the deliberation component, that you wouldn't see it in this area. Whether you would see it during execution, you don't see them. I'll show you. You don't see them during execution. You certainly don't see them transiently during execution. But in fact, you see, as I said, these ramp cells during the execution, and those do occur as the animal's going. So it's possible that there's almost certainly reward information, but I think it's closer to a value type information rather than an expectation of reward. And I'm not going to show that data, but it's hidden on this laptop somewhere. I could find it. Okay, so this is where, don't worry about this. This will come in later. That's an animation that hasn't appeared correctly. Neil Schmitzer-Torbert, this is Neil Schmitzer-Torbert's work. And what he did is he trained animals to both do this multiple T task and then this, what I call a non-spatial spatial task. We actually call it Take 5 because it's in 5/4 time, which is an American jazz joke. So what we have here is a big rectangle with 4 reward sites. And if the animal starts here, he has to go 5 steps around this rectangle. 1, 2, 3, 4, 5, and then he gets food. Now he's here. 1, 2, 3, 4, 5, and now he gets food. Now he's here. 1, 2, 3, 4, 5. And so the point is that space is no longer informative of the correct action. And when we look at the dorsal striatal cells and we ask, what is the spatial reconstruction? And this R squared is a relatively complex, but basically a self-consistency term. What we find is that on the multiple T, there is a good decoding, and it's the slope that matters. As you increase the ensemble size, you get better decoding to space. But as you increase the ensemble size on this other task, this non-spatial T5 task, there is no spatial information at all. None. Now, both tasks has information about, when you get reward. But the space is informative here, but not here. And so Josh Burke has similar results, and Graybill has similar results. In the dorsal striatum, you only see information represented if it's reward informative. If we do this same analysis on, again, a slightly different task, this is a 2/3 task, which was done by Andrew Wickenheiser. The animal sometimes had to go one step, and in this case, we were forcing the animal, so it's a different task. And sometimes the animal had to go 2 steps to get food, right? So here it's processing around. You actually get better spatial representations in hippocampus. So when you take the task and it becomes non-spatial, striatum stops representing space, but hippocampus represents space even better. I think that's a very interesting point. Now, I could show you lots and lots of cases of examples of sweeps that don't occur in the dorsal striatum, but of course, absence of evidence is not evidence of absence. So what we did here is we asked how much of the decoding probability is ahead of the rat and how much is behind the rat. And what's important here is the hippocampus starts out with this kind of vastly ahead and then comes back. This is the same data we were looking at before, just analyzed differently. And in the dorsal striatum, that's not there at all. This is just a constant. It doesn't change. And so what this tells us is that the dorsal striatum is not showing these vast forward sweeps. It's somehow about where I am right now, which makes sense. As I said, there are reward cells in the dorsal striatum, but unlike the ventral striatum, where these reward cells fire extra activity at the 4th choice point, the dorsal striatal cells do not. So again, there's no reactivation of these reward cells at this 4th choice point. There's no difference. Now, some of you may notice this very interesting increase in the non-reward cells occurring in the dorsal striatum. What it turns out these cells are is cue response cells. As the animal comes around this corner, he turns the corner to get food, he hears the click of the feeder. The solenoid triggers, the food comes running down, and he gets it. This is the trigger on this turn, and this is the trigger going from here to here. These are cue response cells that develop within a single day. And they develop slowly over the many laps as the animal automates its behavior.
How many trials do you need, usually?
Before you can actually see those?
Yeah. So when they start to asymptote in the response?
That's a great question. I don't know the answer to that. Less than 30.
Okay.
No, it's a good question. We never explicitly looked at that, but I can think about, we do have it in this 2010 paper, we do have the plot of how it asymptotes about 30 trials. It takes about 20 to 30 trials, whereas the hippocampus goes away in about 10. It seems to be that on this task, the first 10 trials or so, this is a very easy task, because in this task, there's really only 2 choices, go left for food or go right for food. And on these days, and every day, it's once you know it, it's constant. So it's simply on a given day, is it going to be a left day or is it going to be a right day? And so on this task, the hippocampus and ventral striatum last about 10 laps. Then they go away. And then there's about 10 laps where the striatum is still developing. The hippocampus and ventral striatum kind of become mostly only the direction you're going. So in the first 10 laps, the hippocampus is going in both directions, to the left and then to the right, back and forth. In laps 10 to 20, it kind of only goes in one direction. And then by about lap 30, it pretty much goes away. And it's just little bits of what we would see as phase precession locally. And then the dorsal striatum, that's about when these things develop. And the asymptote, they're really key up at 30 to the asymptote probably between 30 and 40 on this task.
But would you expect that there's a dependency here, in the sense that you first want to see some sort of asymptote in the hippocampal ventral striatal learning, after which dorsal striatum starts to kick in?
That's right. That's what we think is going on here, and that's kind of the consistent in terms of these typical behavior. You'll see a deliberative, you learn the map, and then you say, I have to figure out what I'm going to do on this map. And so that takes deliberative behavior. That takes a few laps. You can do that early because it's very flexible. And then as you're doing it over and over again, you automate your behavior. Now, we know from hippocampal lesion experiments that you can actually show automation of behavior without a hippocampus. The problem is you have to find a way to have the animal actually get the right answer, right? So there's the wonderful experiment from Day, Shallert, and Wiegand, there's a middle author whose name I don't remember. Day is the first author. And what they did is they did a Morris water maze task where they had these platforms of different sizes. So they start out with the big, big, big platform, and then they shrink it. And over the course of a month, basically, you can teach the automation behavior in the rat to go to that location, right? So at the end, they look just like, you know, classical animals on a Morris water maze. But actually what they've learned is they've learned at every point what's the cube map that I'm going to go to. And there's a wonderful, and it's hidden in like a middle paragraph somewhere of these results that I think should have been basically the title of the paper, but it wasn't, is that when you shrink the platform, the hippocampally lesioned animals are better than the hippocampally intact animals. Because what the hippocampally intact animals do is they go to where the platform was and they look for it. And so they actually waste time looking for it, whereas the other animals just go and they then find the platform. So we know that these are different systems. They're separably learnable completely. I mean, in some sense, the HM story about you can learn, you know, is separable. So now the question comes one of time. So I can kind of throw in my one conclusion slide and we can stop here, or if we have time, I can go through the coda. What's your time plan?
Well, we have about 10 minutes
Okay.
in our schedule, so.
Yeah, we certainly, this is usually about 10 or 15 minutes.
Okay.
So I'll go through it, because I love this data. This is, to me, some of what I think is the most exciting things we've been doing recently.
You tell us now?
Yeah, that's right. Well, I wanted to, it's the conclusion, right? It's the grand finale, right? All the fireworks are exploding.
Okay.
So let's come back to this story, right? This story of we have some behavior, we have some encoding, we do some decoding, we do the generative models to kind of measure this model structure, is not dependent on space. We've spent all this time doing it spatially, but there's nothing fundamental about space in this. We could do it with any parameter. And so what I'm going to do now is I'm going to use this in a non-spatial domain. In fact, I'm going to use it for flavor. So again, just as we did with deliberation, we have to be careful. What does it mean to regret something? What is regret? Turns out regret, if you go to the psychological literature, regret is the recognition that you made an error. And yes, this is my favorite regret. I assume everybody, even in Europe, gets this joke, right? This is the stormtrooper saying those were the droids you were looking for and regretting the moment. It's my favorite regret picture. So that's right. So basically, if we think about it from a kind of state space type diagram, we have some cue, we take some action. Regret is the recognition that there was an alternative action that would have been better. And that means we want to differentiate regret from disappointment. And disappointment is the recognition that the world's just not as good as you liked. It's not your fault. Regret means I screwed up. Disappointment means, damn, that sucks. Right? And so this comes to the question of agency. Regret requires an agency and counterfactuals. So it needs an economic task. And what I'm going to show you today, to be very careful, what I'm going to show you is that we can prove that rats recognize their own agency. They recognize the difference between this condition and this condition. And in this condition, and only in this condition, they actually represent the action they should have taken. So we're going to do this with this restaurant row task. So the rat is running around a circle. Actually, I think he's going this way, but it doesn't matter. And as he passes, there's 4 spokes, each of which are leading to a different flavor of food. Think of it as a different restaurant. And there's a cherry flavor and a banana flavor and kind of a plain, unflavored, and a chocolate. And it's American chocolate. It does not smell very good, so don't be surprised when they don't like it. I've never tried it. I told my students not to taste it, but Adam says they taste terrible. As the animal runs by one of these zones, he'll hear a tone, and the pitch of the tone will tell him how long he will have to wait for the food. And the pitch will start counting down, beep, 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 by tones, once per second. And the key here is that the animal then has a choice. It can say, I'm going to wait that delay and stick around, or I'm going to skip it and go on and try my luck at the next restaurant. Now, the important point here is two very important points. One, they don't know what the next delay is when they leave the first one. Okay, the delays are random with a uniform distribution of about 1 to 30 seconds. For some of the rats, they were willing to wait 30 seconds. We upped it to 45 seconds. But basically, it's a uniform distribution of delays. The second is they have a limited time on the track. They have 60 minutes to gather all their food for the day, which means that time spent waiting for banana is time you cannot spend waiting for cherry. Go ahead.
I couldn't tell from the paper. Does the tone, something about the tone that tells you
The pitch.
how long the delay?
Yes, the pitch.
The pitch will start higher.
Correct.
If it's a long delay?
Correct.
But then
And then it counts down. So the pitch of the tone tells you the delay. And in fact, one of the things that's interesting is it turns out these rats have actually, coming from Stefano's actually talk, the pitch of the tone, the animals know that the pitch to delay transition, because when we went from 30 seconds to 45 seconds for those animals, those two animals, they immediately got the 45 seconds. And their behavior immediately tracked the higher pitch. They recognized, they were able to translate that. Now, whether that inference, I'm not going to defend logic versus not logic. I don't want to get into that argument again. But it's clear that they understand the relationship between pitch and delay. Go ahead.
One tiny little clarification on that.
Yes.
It always comes down to the same line.
Yes, that's correct. That's correct. It always comes down to the same last pitch. Now, excuse me, a very important question, which is I think what you're actually asking, is are they just waiting for some tone? And the answer is no, they're not. They actually understand the tones. And the reason is because this is a plot of delay by the duration weighted and basically a histogram. And you can see a big plot on the x equals y line where basically they've weighted out the delay. And when they don't wait out the delay, they leave after 3 seconds no matter what. So they're not waiting for a given tone. They're basically making a stay-go decision at 3 seconds, within the first couple of seconds.
Why are you saying that? I don't get that from this.
The important point is that the leave, so that this is bimodal. Basically, they either leave after 3 seconds, that's this bump here, or they wait the whole delay. If they were waiting to say, I'm going to wait to the 10-second tone and then I'm going to skip it, there'd be stuff up here. And that's not happening.
Oh, right, okay.
They do show revealed preferences, because what they do, which is of course what you'd expect them to do, is when it's really low delays, they stick around, and when it's really high delays, they leave. And we can measure the threshold either by a sigmoid or by a heaviside. These are all sigmoids. And you can measure the half point of the sigmoid, which is basically their preference for that food. So this rat would wait 5 extra seconds for the unflavored food than for cherry or banana. This guy apparently didn't like cherry or banana, liked the unflavored food. And this is consistent across days. This is the error bars, the mean and error bars across 30 days for 4 different rats. This rat really likes the unflavored food. This rat hates the chocolate. Okay? So the key here is, first of all, this works. This suggests that we have face value for this task from an economic perspective, which is very exciting. And second, it means we can define good deals and bad deal. A good deal is one that is less than threshold for this flavor, and a bad deal is one that is greater than threshold for this flavor. So this is a very bad deal, 30 seconds, and this is a very good deal for 0 seconds. Okay? 10 seconds is a much better deal on the unflavored than it is on the cherry. And so, again, the way this starts is, we're running some pilot data, and I'm saying that looks interesting, and the grad student comes in and says, Dave, my rats are showing regret. And I say,
You're fired.
Don't do, no, I don't say that, to be fair. I say, rats don't do that. You're going to have to prove it. There's no way. He says, no, but just look. So this was the pilot data that started us off. And I should mention, this is all Adam Steiner's work, which recently came out in Nature Neuroscience. So this is a pilot data. We didn't have the chocolate yet. It's cherry. He thinks it tastes like strawberry or smells like strawberry. I don't know if you guys in Europe have this. It's this cherry flavor they put in kids' medicine. It's this fruit flavor, which kind of, that nasty pink fruit flavor, right? He thinks it's strawberry. I think it's cherry, whatever it is. It's that fruit flavor they put in kids' medicine. So you have this strawberry or cherry flavor, this banana, and in this case, we had 2 unflavored food pellet sites. And this guy is going to encounter 20. He's running this way. He's going to count 21 seconds on banana, 10 on the first, and then 28 seconds on the unflavored. And I think you all can expect what's going to happen. The rat comes through. He looks at the 21 and banana, says, forget it. Notice this is a vicarious trial and error event, interestingly. But he says, forget it. I'm not sticking around for banana. I'm going to go up. And he does a VTE here. And I don't know why he doesn't stick around for 10 seconds here. 10 seconds is well below his threshold for this unflavored food pellet. But he doesn't. And now he hits the 28 seconds, and he's screwed up. He could have gotten food after 10 seconds, and now he has to wait 28 seconds to take it. And he stops, he looks backwards, and just anthropomorphically, he looks frustrated. He looks angry. Okay, so that's all fun. Let's look at the actual data. So in order to do this, we're going to have regret is the recognition that another option was better. So we're going to take this task, and we're going to analyze it by rotating it. So we're going to talk about the current event, the current reward site, which has an entry point into the box, which is when the rat is informed of the delay. So the rat now learns that delay is greater than threshold. And a regret condition is the one where he skipped a previous site, did not take the food, but it was a good deal. So if it was a good deal and now it's a bad deal, he's messed up. He could have gotten food for a better deal, and now he's in a worse deal. This is his fault. He made a mistake. We can control for this with the 2 disappointment conditions. One is he took the previous food, which controls for the thresholds, so the thresholds are going to be the same. And the other one is, of course, that he didn't take the food, but it was the right choice, and the world just sucks. He's kind of really, the world's really bad right now. He kind of had 2 bad deals in a row. And yes, we could think about going 3 and 4 steps back, but honestly, these events are so rare, we haven't been able to analyze it. So we're only going to look 1 step back. Does this make sense to everybody, how this is a regret condition? Because he messed up. There's an agency error here. Whereas here, he did not mess up. Now you could, right, so both of these are correct.
Except for one thing there.
They have 60 minutes, right?
Correct.
So I could also interpret this by saying, well, I'm in this maze for this restaurant carousel for 60 minutes, so whatever, I get another chance.
Except they are very clearly, that time limit is obviously, it's like the pitcher's mound in American baseball. It has, over the course of generations, it moved to be exactly so that the timing of throwing from the pitcher to the batter is just about the right time for somebody to steal a base. We moved that time so that if it's too long, absolutely, they don't seem to care. And if it's too short, they get terrified and they take everything, actually, which is interesting because that's actually not the thing to do. But they actually do worse when you kind of really shrink it down. They actually take just every deal. And 60 minutes turns out to be the perfect balance where, in fact, it is quite clear that they care about these things. Go ahead.
Always trials with behavioral sign of regret? I mean,
no, I'm going to show you. So I'm actually not doing behavioral sign of regret. What I'm saying is I'm going to take all trials that are regret-inducing by this definition. And then what I'm going to show you is that there's a behavioral consequence which shows that they recognize this regret-inducing situation. And then I'm going to show you a neurophysiological consequence showing that there's differences. So I've actually defined the trials from the economic and psychological definition that should create regret. And then I'm going to ask, is the behavior? So the behavior is the question, not the definition. Does that answer your question? Yeah. Okay. So, in fact, this is the behavior. What we see is, so this is, as I said, we've rotated everything so that this is the current site, this is the previous, and this is the next. On the regret conditions, the animals come up, they hear the moment, and they tend to stop and look backwards. They have this kind of stop and kind of, whoa, backwards look. On the first control condition, which is took the previous deal and now have a bad deal, they tend to do a vicarious trial and error looking in, but they don't take it. It's a bad deal. And then they kind of tend to go into the next site. Interestingly, on the second one, where they've had 2 bad deals in a row, they often actually will skip the bad deal and then do the vicarious trial and error coming back and often kind of stick around for it, which is interesting. But we can quantitatively measure this direction, and we actually got the way to measure this actually from a curvature measure from from this computational paper of medical researchers looking at retinal tortuosity of the retina, of blood vessels in the retina, which turned out to be this wonderful equation for tracking kind of when you get these events. And it turns out to work very, very nicely. But quantitatively, you get an increase in looking backwards in the regret condition, an increase in looking towards the current one in this first, and an increase in looking towards the next in the second one. And these are, of course...
How should they interpret this plot exactly?
So this is taking this moment at this, what is the sharpest point of the direction? Take that as the direction of kind of look and stop.
So relative to the main trajectory or something?
Relative to, no, actually relative to this triangle here. And so we've cut it into these three zones. This zone is looking backwards. This zone, this third, is looking towards the current reward site, and this zone is looking towards the next site. So this is the previous, current, and next. The gray lines are the full distribution of all our data, and the red or blue lines are the vector sum of the vectors within that zone. And so what we see is and we can use U-squared measures to differentiate these. And you can see...
Would it have helped you to do it more relative to the trajectory as opposed to the geometry of the task itself?
It might have. I like doing it relative to the geometry of the task itself because it's kind of clean. And measuring the trajectory, you have kind of issues about how noisy are the trajectories themselves. But in any case, we could have gotten better, but we have a significant effect here, so it's only going to make it better, not worse. So what we have is and these are different significantly from each other. The second piece of behavior is that animals are more likely to stick around on the regret condition than on the first control condition. And actually, it's a 0.06, but of the second control condition as well. So they are more likely to take the bad deal after having screwed up on the previous one, which again is something that humans do when they show regret. One of the consequences of regret is you tend to kind of take the next bad deal. Okay, and then the other thing, and actually behaviorally, the thing that convinced me we had something really intriguing here, is that there's a very interesting handling time event, which is that after they get the food, anybody who's actually run rats on behavior knows that they tend not to just run off to the next thing. They tend to sit around, they groom for a while, they kind of, spend the time eating, they look around, they go into sharp waves, they do lots of these things. And typically, if we look at the distribution excuse me, of all the non-regret conditions, we see basically this nice Gaussian, there's a little blip here, but nice Gaussian in about 30 seconds. Take all the regret conditions, they've eaten the food in 5 seconds. So they come in, they take the bad deal, they wolf the food down, and they say, forget it, I'm going on. And that is, of course, very, very different from each other.
There is no relationship with the previous reward received.
This difference?
I mean, on the previous trial.
Honestly, no. This is the difference on the previous trial. That is, in the regret condition, in the two control conditions are less than the regret condition. That controls for both having taken the food previously and for the delay on the previous trial. Because one controls for the behavior, the other controls for the cues. Obviously, we can't control for both because then we'd be in a regret trial again. So, so what we have, as I said, these cells in both orbitofrontal cortex and ventral striatum both respond to reward. Interestingly, not surprisingly, they have different responses to the different flavors. So this is measured at reward receipt. So 0 is the time they actually get the food. This cell likes kind of chocolate, maybe a little of the banana, less so on the, this is exactly the kind of plot that Stefano was showing from his kind of analysis, right? You have basically the peri-event time histogram and the individual spikes underneath it. This cell really likes the cherry in the ventral striatum. Different cells like different flavors. It's not the value per se, it's the flavor that's driving this. But what this means is we can do this decoding story, right? We can say we're going to take our condition, our spike trains, our flavors. This actually shouldn't be labeled in terms of current next. It should be labeled as banana and not, because we're actually we do our decoding in flavor and then we rotate for the analysis. So we have 5 conditions, the 4 flavors and basically the average firing rate and the rest of the maze. That gives us kind of a, well, it's not about reward right now as a 5th condition. We take a new set of spike trains and do decoding. So if we're actually, in this case, we're actually saying, given the reward site response, what's the firing at the zone entry? And what we see, not surprisingly, is at the moment of entering a zone, you see a very good representation of your current zone, of the current reward you expect to get. Not surprising, these are the kind, I mean, this is the kind of data you would see, that you see a cell that responds to reward tends to have some response at the Q that tells you you're about to get the reward. An interesting note, just, I have to say, I didn't even notice this until I was writing this talk for this, and we're now going to go back and look at this. Orbital frontal seems to be preceding ventral striatum here, which is intriguing, given what we saw earlier. I don't know what to make of that. Honestly, I did not notice that until I was putting this slide together. So we'll have to see what we make of that. It's very interesting. Yes. The variation, that is, there is some bump in the variance as well. So the first thing we thought of is maybe there's going to be a representation of the reward that you should have gotten, right? This is what you think of when I think of regret. When I say, well, I should have gotten the previous reward. There is some little bump maybe hidden in here. You know, I wouldn't want to write about it. It's not really there. But then we started looking into the psychological literature. And what the psychological literature told us is you don't regret the thing you didn't get. You regret the thing you didn't do. It's all about the action. It's, I should have done that. So what we did is we said, let's take, instead of our training as the reward site, let's take our training as the entry moment into the restaurant. And now we're going to take our decoding. And now, of course, we have to do a leave one out analysis. But yes, you have a very good representation of the restaurant that you're entering, the zone that you're entering. So this is probability of zone given spikes at zone entry. And here you have a huge response in the regret condition of the previous action. Now, that's not, in the first control condition, you don't have any of that. What you see is a representation of the current one which says, forget it, it's not a good deal, I'm going to think about my next option. There is some representation of both the current and previous, but they are not significantly different from each other in the second control condition. So if we want, the only one that has a clear representation, and it is highly significant, of the previous site that's different from all the others is the regret condition. In the regret condition, there is a representation of the thing you should have done. Go ahead.
I presume we're showing individual neuron results.
This is the decoding. So this is the probability of decoding to each of these sites. This is the decoding probability. We're working in information space, not neurons.
So could you give us a little bit of detail on how decoding is done?
Yes,
in terms
it's
of what the population
it's the Bayesian decoding. So you take a population of neurons.
Roughly how large?
50.
Okay.
So you have a population of about 50 neurons. Actually, I think it may even be less because it's split between 2 structures, but tens of neurons, you know, multiple dozens of neurons.
Okay.
And you take the you take the training set, and that says that's P of X given S, right? Where you have, and we're talking firing rate, because we take the 3 seconds after, either after reward site or after zone entry, obviously cutting it at the time he actually gets reward in the couple of cases where it's less than 3 seconds. But you take the 3 seconds, that gives you a number, that's probability of X given spike, and then you do the Bayesian inference. And so at every moment in time, from a current population of firing, you have what is the decoded probability that this representation is encoding the different sequences, and that gives you a function as a function of time.
And presumably the prior, which prior do you use?
We're using a uniform prior over these 5 conditions. So it's 0.2.
Okay, so they're completely independent.
Yes.
Okay.
But is your interpretation then correct to say regret is what you should have done? This data would show that the regret is about where you should have been.
Sure. And in this case, so we cannot differentiate between the action at that location and the location at that moment. I agree. And I think from the orbitofrontal representation, it's kind of, I wish I had been at that moment in time. And what we would need to do to get to action is actually have cells that represent the different actions at that moment. And no, you're right, we don't have that. What we have in the orbitofrontal is the representation of that episodic moment.
Exactly.
And then we go back to that.
But you could also argue regret is a relative notion for which you need a reference, and the reference is that previous zone.
Yes,
right?
That's right.
That would be then the point.
Well, the point is that what regret entails is that counterfactual moment.
Right.
Right. And that's what we're seeing, is we're seeing going back to that counterfactual moment. So what's really going on here? I mean, we talk about this in terms of information space, but fundamentally we're talking about neurons. So this is what's driving that event. To be honest, I don't like thinking in these terms because I think it's too easy to find specific examples. I think this is the way to quantitatively analyze the data. But for all of us who live in neuroscience, you really want to come back to the cells, what's going on. So here's a specific example. The rat has skipped a 8-second delay, even though his threshold at cherry is about 18 seconds, and has now encountered a banana where his threshold is also about 18 seconds with a delay of 25 seconds. So this is skipping a good deal and finding a bad deal at this moment. He stops, looks back, and this cell generally fired its massive amount of spikes on entry into the cherry zone. When it enters into the banana zone, there's never any firing, except this one time we get a burst of firing that, if you look at the firing rate, basically tracks this approximate 8 hertz. So what we're seeing is cells firing at the wrong time that are consistent. Now, this is one cell. Obviously, we'd have to look at lots of them, which is why you do this. And what you say is the population of cells that are firing are actually more consistent with that other moment in time. And the nice thing about the reason I like this is that this is not space. I mean, yes, there's issues of is there actually a spatial component to this, but we're doing the decoding by flavor, by restaurant. Now, does this actually correlate with the behavior? One of the things that's interesting is we then said, let's take the difference between the representation of the previous restaurant entry and the representation of the current restaurant entry, and there is a, well, I would call that significant. I conclude all the numbers. You can decide what you want, where you want to put your thresholds. There is a difference in orbitofrontal and ventral striatum between sticking around for this bad deal and skipping the bad deal, only in the regret condition, not in either of the two control conditions. That is, the neurophysiology is reflecting the subsequent behavior that he's going to do in this regret-inducing condition. So what I've tried to argue today is as a way of analyzing data, a way of doing neuroscience, by taking psychological terms, deliberation, regret, getting that information processing, saying what is the actual information processing that a regret event is going to require, that a deliberation process is going to require, and then taking the algorithm, what is that computation? That creates a neural structure question that we can go in and we can look and we can say, is that information processing happening at the right moment? And then, of course, we link it back by asking, is that information component work? And obviously, these goes in all directions, and it's, you can, this is a way of thinking about connecting the psychology and the neuroscience through computation and information processing. And I believe the kind of best way to do that is this loop. And as I said, the key is, when does this model require the addition of a covert component? When do you have to say, there's a replay event happening? There is a moment of representation of the other option at this moment. And that comes in by following this loop. So I will end there, except I want to acknowledge a few things. First of all, I want to acknowledge all the students. I tried to go through very carefully and identify the graduate students and postdocs who did the actual work as we went. I want to acknowledge the funding sources, both NIH and NSF and HFSP. And I want to take a moment to acknowledge my rats, because they're willing to work for us. They're willing to stop and wait and think for us. And that has, as Robert Frost would say, made all the difference. Thank you very much.
All right, question time. Caesar.
About the regret condition.
Uh-huh.
Sometimes you also regret because like you make a bad decision and then suddenly you're faced with a better deal. And
You weren't the reviewer on a paper, were you?
no.
No, he's always.
I happen to know who the reviewer was, so but because he told me afterwards. One of the reviewers asked exactly that point, and I was like, "Oh, forget it. There's no way this will work." Except it turns out that in the condition where you take a bad deal and then hit a good deal, you also see regret. You also see a representation of the previous site. I was totally shocked, but it's absolutely there.
So you haven't mentioned consciousness yet.
No, I haven't.
But it seems like you have the only data relevant. I mean, so when the rat chooses at the choice point and you play out both sides, isn't that consciousness?
So I used to actually, put a little joke in when I'd show the vicarious trial and inner deliberation data and say, well, neither I nor Tolman would ever use the word consciousness here, having, of course, just used the word consciousness. Except after I did that and Adam was in the audience, he said, well, have you read Tolman's 32 book? And I said, no. And he said, well, go read it, because chapter whatever is called Vicarious Trial and Error is Proof of Consciousness in the Rat. So Tolman would say it. I'm not comfortable with the word consciousness because I have no idea what the word consciousness means. My opinion on the word consciousness is that it's folk psychology, and I don't want to go there. To me, the question is, what's the information processing that's happening that we actually tend to associate with this consciousness term? And I'm very comfortable with free will. I believe my rats have free will. I think that they have agency, right? I think we're seeing agency in the rat. I'm very comfortable with the word agency. Consciousness, I don't know where to go. We can talk about it over a beer, but I don't really know what the word consciousness means. So I'm not comfortable using the word, even if Tolman was.
But it seems interesting to then equate free will with agency, because they're different things. I mean, agency just means that there is a knowledge of causal relation to the world, right? Well, free will is actually the source of that causal relation.
I agree.
Okay, yeah, we should have a few beers on that. Are there other questions?
I think the key is agency.
Yeah.
And
no, because
agency is a much better term for this.
Sure. But the other thing is regret. Now, looking at your data, do we really need to use the concept of regret, right? Because you could also, let's say, deconstruct it in terms of, let's say, a re-evaluation of an expectation, right? That we can sort of tone down a little bit all the psychological overtones.
Right. So there is a very important question about whether, so the reason I bring up, the reason to me the regret's the right term is that I want to differentiate the recognition of its own agency relative to disappointment. So under disappointment, you also have reevaluation of process. I mean, that's the whole dopamine story, right? So it's not just reevaluation of your previous events. There's a separation between the agency component of what condition A and condition B. And under the psychological terms, that agency differentiating the psychology literature, which there's a list of names that are escaping me, particularly this one Dutch guy whose name I don't remember. But anyway, there's a number of psychological people who've been kind of defining regret, and the key has been that agency.
Right.
Now, there is a very interesting question about affect, which is when we feel regret, there's an emotional affect component to it. And I have no evidence that the rats are doing that, because honestly, I don't even know where I'd find it.
Right.
I suppose we could look at kind of frustration behaviors and see if those frustration behaviors are different. We haven't done that. Anthropomorphically, it looks like, I mean, anthropomorphic is the wrong word. Anecdotally is the word I mean. Anecdotally, we do see more frustration behaviors, and that is, because we watch the rats. Right. We certainly, they seem to gnash their teeth. They tend to kind of, as I said, they turn back, But we haven't quantified that. My hope is we'll be able to start trying to find some sort of fundamental, the equivalent of a GSR that we could differentiate, but we don't have it yet.
But then I could still argue, certainly from also the tradition of Tolman and Hull, that the distinction you now make for regret is actually a distinction in Pavlovian and instrumental training procedures.
No, it's not Pavlovian instrumental. This is all instrumental. And actually, the Pavlovian...
Well, maybe not in the regret case, or let's say in the disappointment case, because in the disappointment case, the world is just different than your...
No, he's still making actions, and it's actually, in this case, the offers that he's getting are different. It's still, this is all still instrumental. In fact, given the vicarious trial and error events, this is almost certainly all deliberative. He actually does not seem to automate these behaviors. And one of the points of the book, since I've got,
Which book is that, David?
That's right. This wonderful book that should be required reading in every class is that one of the points of the book is that the way to think about decision making is in terms of these different systems, including Pavlovian, deliberative, and instrumental, and we can use the kind of information processing to differentiate them.
Okay.
And under that differentiation, the restaurant row task is all deliberative, very clearly.
So I should start a review of your book then.
That's right.
John.
I get to maybe make the last comment. Brandeis, we did a journal club about this paper on regret, and at the end, the speaker took a poll about how many people believed.
And how many people regretted reading the paper.
They were all looking behind them.
That's right.
And so about 50% of the people, accepted it. And I think the people who did not accept it were questioning the role of of emotion. So when you think of regret, you do think of an emotional aspect. But what I think is really interesting, especially your last comment, is that that additional step can now be taken. So, I mean, if you can find an emotional correlate, I think that would finish the story. And it could be a cortisol change, or a blood pressure change or eye dilation change, any of the things that we might more accept.
We do have an ultrasonic microphone now. So
there you go.
We don't have data on it yet, but we do have we bought an ultrasonic microphone for exactly that question.
Yeah.
I would love to see the rat curse. But as I said, we don't have any data on it.
You mentioned that the rats are your collaborators, so there is no behavior without their collaboration.
Correct.
Do you ever observe they feel that you are unfair to them?
Yes.
So not only they regret, whatever, they're disappointed, but unfairness, animals are very sensitive to it.
So again, anecdotally, training an animal is a very careful balance between making the task too easy and too hard. And animals will, if you make the task too hard too quickly, they will in fact just say, they'll stop working. And they'll kind of kick into a, I'd rather starve than do this mode. And that's a terrible thing, and then you kind of have to kind of get them back into shape. And there is definitely this balance of making sure that the task gets hard enough at the right level. I mean, it's a teaching issue, and you see that just like teaching, students, right? You have to keep the difficulty kind of just a step ahead of where they're at, so that they'll keep going. If it jumps too far, then they just give up. And so I do think that there's kind of that fairness level that they care about. I don't have a way of quantifying it yet, but it's worth thinking about.
Ivan.
I don't know, it's speculative, so maybe I'm very wrong. But isn't it like that both regret and disappointment share a common computational aspect of reevaluation, but then regret has on top of it something that it's maybe non-functional? It's like a response that it might not be useful.
This is part of why I went back. We go back to the psychological literature on the terms. The reevaluation terms in the psychological literature is based on agency. And so regret certainly has, regret is a recognition that you made a mistake and should not make that same mistake again. One of the things that I would like to see, and we don't have enough samples, I think, in this data set to really see it. Obviously, we're pursuing this this restaurant row task much farther now. But I would like to see if after a regret incident, do they change their behavior subsequently? We know they have an immediate response that's different. But do they actually reevaluate? Does the threshold shift, for example? We don't have enough data to see that. So I don't know if it does or not. But that would be a very interesting question. Does it shift overall? Does it shift for this case? You know, there's a lot of such questions that we could look at to see that reevaluation. We don't know that at this point.
David, have you checked about maybe a notion of urgency on this thing? For example, if you do in the beginning of the task, maybe, okay, you don't regret that much, you still have a lot of time. But maybe like you have just 1 minute left, then you can have maybe some kind of modulation on much of the recall.
We did look over the course of the task, and I think at this point the animals know by this point they've run this task a lot. They've run at least a month on the task before we start recording, and then we run a month of recording to get enough data. So they know the task really well. I kind of think they're more, I don't think, we have not seen anything that changes over the course of that 60 minutes. So that suggests to us that by this point they kind of know, okay, I'm in task mode, you know, and they're ready to roll. Whether we do see kind of in some early behaviors, we haven't studied them, that there are kind of definite shifts, you know, as they, you know, this was, you know, for example, we were saying, you know, if you have too long, right, they satiate. They say, I'm full, I'm done. And actually what's interesting is they go into more deliberative mode. So in the habit cases, if you look at when the vicarious trial and error happens, you'll see it on the very first lapse and occasionally on the very last lapse. And what'll happen is that they'll automate, they'll basically get full, they'll sit for a long time, and then they'll go exploring. And when they go exploring, they really kind of do anything. So I definitely think you can see that. But again, we try to titrate our tasks to get rid of that last data set, that last, you know, exploring component, because that really kind of complicates the system. But anecdotally, we definitely see satiation. But on this task, we've again titrated it, so it's not, 60 minutes is about what they need, and they don't want to waste any time at any part of it.
Great. Well, so let's thank Stefano and David again for a great morning show.
Thank you.
Thank you, David.